Welcome to the deep dive. Today we're tackling the really compelling story of polio.
Yeah, it's a big one. For generations, it cast such a long shadow, didn't it?
It really did. So we're going to get into its history, the huge impact it had, and, well, the amazing science that eventually helped us get a handle on it.
And we know you, our listeners, are curious about these big moments in medical history, so we're excited to unpack polio.
Absolutely. We've pulled together quite a range of sources for
We
this.
have. We've got historical medical texts, the first real descriptions. Then there are the scientific papers digging into the virus itself,
Uh-huh.
and reports from the big players like the CDC and WHO tracking the whole eradication effort. It's definitely a story with many layers.
Exactly. So our mission here is to pull out the most important, maybe the most intriguing insights. We'll follow the thread from those earliest descriptions,
Way back.
right up to today, with things like synthetic biology even playing a part. Okay, so let's start at the beginning. How did we even recognize this thing?
Well, it's funny, polio feels quite modern in some ways, but descriptions of similar illnesses go way, way back.
Right, like that Egyptian stele, the one with the figure who seems to have a withered leg.
Exactly. Could be polio, could be something else, but suggestive. But for, let's say, more formal medical notes, a key name is Michael Underwood, back in 1799.
Underwood, yeah. He was quite established, wasn't he?
Yeah.
Royal College of Physicians, physician to the Princess of Wales.
Right, and the British lying in hospital. He wrote about various things, ulcers, kids' diseases.
And it was in that children's diseases book, the 1799 one, that he described this weakness, specifically in the lower limbs of children.
He called it a debility of the lower extremities. I mean, reading it now, it's unmistakably polio he's describing.
So the seeds were there, observationally, even if the full picture took ages to develop. Shows how important just noticing things is in medicine.
Absolutely crucial. But, even with Underwood's description, we didn't really see major outbreaks, epidemics, until much later.
When was that?
Late 19th century, really. And then the first half of the 20th century, things got much worse, especially in developed countries, Northern Hemisphere. These summer-fall epidemics, they just got bigger and scarier.
And the peak you mentioned, 1952 in the U.S.
Yeah, 1952, over 21,000 paralytic cases. Just imagine the fear that year.
Unbelievable. So, okay, what is this virus that caused all that? Let's get into the biology a bit.
Right. So polio is caused by enterovirus C, and the way it gets into our cells is pretty specific. It binds to this receptor, CD155.
Also called the poliovirus receptor, PVR.
That's the one. Think of it like a specific docking station on the cell surface. The virus has evolved to latch onto it perfectly.
Like a lock and key.
Precisely. And understanding that interaction is, well, it was vital for figuring out how to block it later on. This receptor, it's a glycoprotein, and the gene for it, the PVR gene, is on chromosome 19.
Okay, so it docks using CD155. Then what? It gets inside the cell.
It does. Once it's in the cytoplasm, the main body of the cell it needs to replicate, and it does that by using a part of its own RNA called the 5' non-translated region, or NTR.
So the NTR is like its instruction manual for taking over the cell's machinery.
That's a good way to put it. Yeah, it hijacks the cell to make more copies of itself.
Now, polio is the famous one, obviously, as part of a bigger family, right? Enteroviruses.
Oh yeah, big family. There are actually 61 other non-polio enteroviruses known to infect humans.
61? Like what?
Things like Coxsackie viruses, both A and B types, and echoviruses. They cause a whole range of stuff.
Some mild, like colds, some more serious.
Exactly. But not polio. It's a reminder the viral world is pretty crowded. Now, something else that comes up is vaccine-derived poliovirus.
Ah, yes, VDPVs. This is really important for the eradication story, isn't it?
Crucial. So VDPVs are basically strains that have evolved from the oral polio vaccine, the OPV.
Because the OPV uses a live but weakened virus.
Right, it's attenuated. But because it's live, as it replicates in someone's gut, there's a tiny, tiny chance it can mutate over time.
And potentially regain the ability to cause paralysis.
Potentially, yes. And if vaccination rates are low in a community, these mutated strains can start circulating. There was a case in Spain, 2008, that really highlighted this.
So it's a trade-off then.
Wow.
The oral vaccine is great for mass campaigns, easy to give,
Absolutely. And provides good community immunity.
but carries this very small but real risk of reversion, especially in under-vaccinated areas.
That's the difficult balance, exactly. Okay, let's rewind a bit. Before the vaccines, what did treatment even look like?
Yeah, before prevention, it was all about managing the aftermath, wasn't it?
Yeah.
A lot of physical therapy, trying to maintain function, deal with deformities,
Yeah.
these traction, things like that.
And managing pain, secondary infections.
Right. And then the iconic, terrifying image, the iron lung.
Oh, absolutely. These huge negative pressure ventilators, life-saving for people whose breathing muscles were paralyzed, but
Imagine living in one. It just screams desperation for a preventative measure.
it really does. Which brings us to vaccination eventually. But even before that, there were other approaches people tried, right? Like thermal baths.
Yes, like in Bath, England. The King's Bath, Queen's Bath, hot bath, all slightly different temperatures. People went there for all sorts of ailments.
And Warm Springs in Virginia, famously used by FDR, similar idea to places like Wildbad in Germany.
Supposedly good for chronic issues, pain. Though they even knew back then it wasn't a cure-all, and maybe not for everyone. Interesting historical footnote.
It is, but like you said, the real revolution was vaccination. How did we get from baths and iron lungs to that point?
Well, the very early attempts using actual infected tissue,
Okay.
that was way too dangerous. Could give you polio.
Right. Makes sense. So the real progress needed more fundamental science.
Exactly. Things coming out of what they used to call life lore. Basically, embryology, understanding how organisms develop.
How does embryology connect to vaccines?
It helped figure out how tissues grow, which was key for learning how to grow viruses in the lab. You needed to study them, right?
Yeah.
So work on tissue transplantation, cultivating viruses, inactivating them. It all built up.
So understanding basic life processes paved the way. It wasn't a direct line, but foundational.
Right. And you sometimes see mentions of figures like Christian Wolff. His philosophy seems a bit removed.
Yeah, Wolffian philosophy. It wasn't about polio, obviously, but his whole approach, like blending logic, metaphysics, trying to systematically understand nature, it kind of contributed to the broader intellectual climate.
Ah, so fostering a way of thinking that encouraged scientific inquiry generally.
Sort of, yeah.
Yeah.
Creating an environment where asking those detailed questions about the natural world became more rigorous, then you get more specific biological insights.
Okay, that makes sense, like the work by Haeckel and Roux.
Exactly. Haeckel, 19th century, meticulously documenting development from a single cell fertilization, really basic biology.
Yeah.
Then Wilhelm Roux, late 19th century, doing these really early experiments, growing tissues, frog embryos in solution, just trying to keep them alive outside the body.
That sounds like the beginning of tissue culture.
It really was. Harrison built on that, growing frog nerve tissue in lymph fluid. Then Carroll and Weller really refined it, establishing the methods for growing cells and tissues in biological fluids.
And that was the breakthrough needed for virology.
Absolutely critical. You need to grow the virus to study it, to produce weakened or killed versions for vaccines. Tissue culture made that possible.
All these steps. Fundamental biology leading eventually to tackling polio. Which brings us finally to Salk and Sabin.
Right, Jonas Salk. Huge figure. He brought expertise from his Army research on viruses and immunity to the National Foundation for Infantile Paralysis.
And his approach was the inactivated vaccine.
Yes, the IPV. He took poliovirus and used formaldehyde to kill it. The idea was the virus couldn't replicate anymore, couldn't cause disease.
But it would still trigger an immune response.
Precisely. It still looked like polio to the immune system. That vaccine was introduced in 1955.
And the famous story, testing it on himself and his family.
Yeah, that's often told, the creation myth part from 1952. And the narrative about him putting it in the public domain, refusing to profit.
Was that true?
Well, the Foundation's stance on patenting might be complex, but Salk's commitment to accessibility was absolutely real. He wanted it available to everyone. A remarkable stance.
Definitely. And then came Sabin.
A few years later, Albert Sabin developed the oral poliovirus vaccine, OPV, introduced in '61. Different approach.
Live but weakened virus.
Exactly, attenuated, given orally, drops usually,
Right.
which made mass vaccination campaigns much easier.
Easier logistically. And it had other advantages too.
Yeah, longer-lasting immunity generally, and it provided community protection, herd immunity, because vaccinated people could shed the weakened virus, potentially immunizing contacts.
So Salk's was injected, killed virus. Sabin's was oral, live weakened virus. Both absolutely vital.
Totally. Different tools for different stages and situations in the fight. Now, Salk's legacy isn't just the vaccine, there's the institute, too.
Right, the Salk Institute for Biological Studies. Founded in the early 60s in La Jolla.
Beautiful place, I hear. Louis Kahn architecture.
Yeah, apparently designed to foster discovery. Column-free labs, that central courtyard. The channel of life leading to the sea of discovery. Very poetic,
Sounds inspiring. And the focus was broad, right? Not just viruses.
very broad. Improving human health through basic research. And this idea of biophilosophy came up around that.
Yeah, around 66, maybe the New York Times coined it, maybe Salk. It reflects that mix of biology and deeper questions about humanity.
So interdisciplinary. They did behavioral science, biomedical research, even contract work for the government like army vaccine production.
A wide remit. And Armand Hammer got involved too.
Yes, he established the Cancer Biology Center there in '69, added a major focus on cancer research.
So polio, then cancer, fundamental biology, quite the evolution. And Salk also wrote a book with his son.
He did, yeah. Jonas and Jonathan Salk, 1981. World Population and Human Values, A New Reality. Looked at population, quality of life, societal structures.
Broad thinking. And Jonathan later talked about early childhood experiences affecting social systems.
Yeah, the neurobiological impact of early contact, things
Mhm.
like that, making those wider connections.
Interesting. And the Salk Institute also became home to the Neural Information Processing System Foundation, NIBA-IPS.
That's right, their office moved there by '94. Shows that connection to computational science, information processing,
Mhm.
again, very interdisciplinary.
Always pushing boundaries. Salk himself seemed interested in those bigger questions too, like conditioning.
Yeah, there's a record of a conversation he had with Krishnamurti in '95 talking about how we're conditioned and the possibility of, well, unconditioning ourselves,
Hmm.
understanding those processes.
It really bridges that gap C.P. Snow talked about, the two cultures of science and humanities. Salk seemed to embody both.
Seems like it. And people linked to the Institute were involved in other things too, right? The Jonas Salk Foundation, Immune Response Corporation. Kevin Kimberlin's name comes up.
Right, a whole network spinning out from that initial work. Now, you mentioned Salk's Army research earlier. Let's dig into the military's role a bit more.
Okay. The U.S. Army was actually hugely important, especially for influenza vaccine work. They set up a specific board for it back in 1941.
During the war, makes
Yeah.
sense, influenza could cripple troop readiness.
Exactly. So early flu vaccine trials were often done on Army personnel, sometimes with Rockefeller Foundation money.
And they were using adjuvants, even back then, these immune boosters.
Yeah, Salk used one called Freund's Incomplete Adjuvant in a 1951 study at Fort Dix. It worked well to boost antibodies.
There's always a but with early adjuvants.
Right. Freund's caused quite a bit of inflammation, pain, serious concerns about side effects, even its humaneness in animal studies later. Effective, but rough.
So modern adjuvants are different.
Oh yeah, much more targeted. We understand toll-like receptors, TLRs, on immune cells now. They recognize bits of pathogens.
So adjuvants trigger those specific receptors.
Many do, yes. It gives a more controlled, tailored immune boost. We still use alum, aluminum salts, but there are newer ones approved too, always trying to balance effectiveness and safety.
That makes sense. And the Army's Commission on Influenza really pinned down the importance of antibody levels, right? Antibody titers.
Absolutely.
Yeah.
Their studies clearly showed higher antibody levels, whether from infection or vaccination, meant better protection against getting sick. That's fundamental to vaccine strategy even now.
Okay, so huge progress, but not without bumps. The Cutter incident is a major one.
Yeah, 1955, just as the Salk vaccine was rolling out, a disaster.
What happened exactly?
Some batches from Cutter laboratories weren't properly inactivated. They contained live virulent polio virus.
Oh, no.
Yeah, kids got polio from the vaccine, some family members, community contacts too. Vaccine-associated paralytic poliomyelitis.
Horrifying. A stark lesson in quality control.
Absolutely critical. Rigorous testing, safety checks at every single step.
Yeah.
Then later, another issue surfaced, SV40.
Simian virus 40. This came from the monkey kidney cells used to grow the virus for some vaccines.
That's right, discovered in 1960 by Sweet and Hillman. And the worry was...
It caused tumors in hamsters.
Exactly. Which led the NIH pretty quickly in the early 60s to stop distribution of contaminated vaccine lots and also put a hold on live adenovirus research because of similar cancer concerns.
And Maurice Hillman, who found SV40, was a giant in vaccine development, right, at Merck.
Huge career. His vigilance was key there. Later studies, like from the Institute of Medicine, tried to see if there was a real link between SV40 and human cancer.
But it's remained unclear, right? No definitive proof of a causal link.
Pretty much. But it shows the level of scrutiny these things undergo, even years later. And we know a lot about SV40's genetics now.
Yeah, the whole genome is mapped. Origin and replication, promoter sites, splice sites, all annotated. That detail is crucial for understanding any potential risk.
Definitely. Okay, so despite these serious issues, the vaccines did work incredibly well overall. Let's talk about the push for eradication.
Right. After OPV came in 61, polio cases in the U.S. just dropped like a stone. The last wild case caught in the U.S. was 1979.
Amazing turnaround. And that led to the global effort.
Yeah, the Global Polio Eradication Initiative, GPEI, launched in 1988 by the World Health Assembly. Massive partnership.
WHO, CDC, Rotary International, UNICEF, all pulling together.
With the goal of wiping polio off the map completely. Hugely ambitious.
But as we touched on earlier, the vaccine-derived strains, CVD-PVs, became a major hurdle.
The biggest challenge now, really. These strains emerging from the OPV in places with low vaccination coverage. They can circulate, regain virulence.
And the rarer IVD-PVs in immunocompromised people who shed the virus for a long time.
Right. So the tool itself presents this endgame challenge. How did the GPEI adapt?
Well, big move was the synchronized global switch from trivalent OPV types 1, 2, and 3 to bivalent OPV, just types 1 and 3.
Because wild type 2 is already eradicated.
Exactly. So that switch removed the source for type 2 VDPVs. But if type 2 does pop up now from vaccine-derived sources,
They can use a monovalent type 2 OPV, MOPV2, just for that outbreak response.
right? And now there's also NOPV2, novel OPV type 2, engineered to be more genetically stable, less likely to revert.
Constant innovation. But we've still seen CVD-PV outbreaks. Egypt in the 80s, 90s, Philippines 2001.
Yeah, and genetic sequencing showed some of those VDPVs had even recombined with other enteroviruses. The virus is tricky, always evolving.
Which leads us into some really modern science,
Yeah.
synthetic biology. What's its role here?
This is pretty mind-blowing stuff. Back in 2002, Eckerd Wimmer's lab actually synthesized the entire poliovirus genome from scratch.
Created infectious virus from chemical ingredients.
Essentially, yes. Based on the known genetic sequence, it showed it was possible.
Wow. What are the implications of that capability?
For research, it's huge. You can design and build viral genomes with specific changes to study exactly how they work.
Like figuring out how the RNA gets packaged inside the virus particle, encapsulation.
Exactly. Or identifying things the virus needs from the host cell to assemble itself, like glutathione for polio's morphogenesis.
And making infectious RNA from cloned DNA in a test tube that allows for really precise genetic manipulation, right? And mutagenesis.
Yeah, change a single letter in the code and see what happens to the virus's ability to replicate or make proteins. A powerful tool for understanding basic mechanisms.
But, I mean, synthesizing viruses, that sounds like it could have downsides too, dual-use concerns.
Absolutely, that's the big ethical discussion. David Evans's work synthesizing horsepox virus, for instance.
Which isn't harmful to humans and might even lead to a safer smallpox vaccine. He made a synthetic chimeric version for that, right?
Correct. But his work also highlighted that maybe synthesizing a virus isn't as incredibly difficult or resource-intensive as people once thought.
So the same techniques could potentially be used for more dangerous pathogens.
That's the worry. It underscores the need for really careful oversight and responsible science in this area.
Definitely. Yeah, other techniques exist too, like heterologous reactivation, using Shope fibroma virus.
Yeah, that's a way to build recombinant vaccinioviruses, often used as vaccine vectors.
Mm.
You use one pox virus to help manipulate the DNA of another. Useful for research and maybe developing new vaccines.
It's interesting. All this high-tech synthesis, but basic viral culture, like the CDC used for SARS-CoV-2, is still fundamental too.
Still essential.
Yeah.
So how do we actually detect polio today in a clinical or surveillance setting?
It's a mix. Growing the virus in cell culture is often the first step to isolate it from a sample.
Okay.
Then real-time reverse transcription PCR is the key. It detects the virus's genetic material. It's super sensitive, specific.
And it can tell the difference between wild polio and vaccine-derived strains, that intratypic differentiation.
Exactly. Crucial for tracking outbreaks and understanding the source. There are also antibody and antigen tests used sometimes.
In cell culture itself, that basic technique is still central.
Absolutely. Growing cells in sterile media nutrient broth in petri dishes or flasks, inoculating with the sample. Using trypsinization to passage the cells, it's laboratory bread and butter.
And looking ahead, there's even cell-free manufacturing, making proteins without whole cells.
Yeah, using cell extracts, energy sources like PEP, DNA templates, you can synthesize proteins directly. Potentially safer for some applications, maybe easier to deploy.
And things like synthetic nanoparticle antibodies, SNABs, are being made that way, as alternatives to monoclonals.
That's an emerging area, yes. Shows how manufacturing itself is evolving.
Incredible. And PCR, you mentioned it for polio diagnostics, but it's huge elsewhere too, like forensic.
Oh, absolutely. PCR combined with STR
Right.
analysis, short tandem repeats is the bedrock of forensic DNA testing now.
Amplifying tiny DNA amounts, looking at those repeating sequences that vary between people.
Right. It's robust, highly discriminating for identification, and importantly, the STRs used are non-coding. They don't reveal physical traits.
And the FBI validates these methods rigorously. It's the standard now.
Pretty much worldwide in forensic labs. Amazing how these molecular tools span medicine, public health, law enforcement.
It really is. Wow. Okay. We have covered a lot of ground here, from the very first hints of the
Right
disease,
back to Underwood, maybe even earlier.
through the terror of the epidemics, the triumph of the
The
vaccine,
setbacks like Cutter and SV40.
to the massive global effort to finally eradicate it. It's quite a story of science, persistence, and collaboration.
It really is. And it shows how progress comes from so many different angles, converging observation, virology, immunology, now even synthetic biology. All essential pieces.
Which brings us to a final thought for everyone listening. We've traced this incredible journey, and we've seen these powerful new tools like synthetic biology emerge. So the question is, what new challenges, ethical or practical, might these kinds of advanced capabilities bring as we continue the fight against infectious diseases in the future?
That's definitely something to think about. Where does this power take us next, and how do we wield it responsibly? A crucial question as we look ahead.